Hello, everybody. Good morning. I'm José Lizcano. Actually, I'm full professor of biochemistry at the University of Autónoma of Barcelona, at the Faculty of Medicine, where I do all my teaching. I'm from, 2 years ago, in June 2021, we moved the laboratory into the VHIR, which I'm very thankful to the VHIR institution because I think it has been a very nice experience. And we are located at the Colserola buildings, and we belong to the eCORE cancer. We are expert in protein kinases, as you may guess from the name of our group. We love phosphorylation, events, cell signaling. We are expert on that. We have the technology and know how to work more than 25 protein kinases, and we offer this technology and know-how to the community. Since we are here, the last 2 years, we have organized 2 international meetings, focusing HIR5, which is a new MAP kinase, but also in the PTK1 AKT mTOR pathway. And since then, one of the main projects of my lab is working in the new MAP kinase HIR5, which is it's meant to play an important role in oncologic process. And because of that, we started, for the last 2 years, we engaged in several collaborations with other groups in VHIR working in cancer. Like we are now studying the role of F5 in neuroblastoma, in endometrial cancer, prostate cancer, or melanoma. So we have different projects. But today, I'm not going to talk about F5, which is my beauty, but I'm going to talk about this little guy, which is called ABTL 0182. From now on, I will call ABTL, or a ver la tele, as we call it in the lab, you know. And this is a very small molecule, and I will talk about the preclinical and clinical development of this compound, because we do collaborations with biopharma companies like Ability Pharma, which is in charge of this product, okay? And this is a alpha-hydroxylated linoleic fatty acid. It's a very slow metabolite. It's a fatty acid derivative. And it's a first-in-class drug, anti-cancer drug, and it has a unique mechanism of action. I'm going to talk to you about ER stress and UPR activation. And now it's in clinical phase 2B for prostatic cancer, but also for endometrial cancer and lung cancer. So, as you know, most of the proteins are folded in the lumen of the endoplasmic reticulum, and to do so, they need the help of other proteins, which are called chaperones. ER stress, ER dilatation happens when there is accumulation on unfold proteins. When that happens, you know, there is a specific cellular signaling, which is called UPR, the unfold protein response signaling pathway, that allows to recover the cell from this stress, the accumulation of unfold proteins in the ER, in the endoplasmic reticulum. And the UPR philosophy is to restore the homeostasis of the cell by halting in general protein translation. There are problems with the chaperones also, so they stop the protein translation. Also, this program and the UPR program started translation of specific protein, which are the chaperones, to help to the refolding of these proteins, and also started the degradation of misfold proteins. That's the normal happening, but if the stress is very robust, very strong, and very prolonged, sustained during the time, the same signal that recover the cellular homeostasis, the UPR, can engage cell death, which is the concept we are working with. So normally, in tumors, under congenital pathways, tumor microenvironments, angiogenesis, many other factors, in cancers, cancer cells live with persistent but moderate ER stress. This persistent and moderate stress allow these tumor cells to proliferate and to evade the immune system. But it has been proven, and our molecule is one of the examples, that you can take advantages of this and induces using ER stress activators to take these cells, especially tumor cells, to cell death, to activate to certain levels the ER stress that turns in tumor cell cancer cell death. And that's ABTL. So To shorten very quickly, because we have no time, 10 years of research in just few seconds, ABTL is induced ER stress and UPR-mediated cytotoxic autophagy in cancer cells, but not in healthy cells. And we have found the ABTL is a very dirty compound, but specifically we have found that ABTL inhibits the saturase 1, which is the enzyme which catalyzes the last step in the synthesis of ceramides, sphingolipid ceramide. So because this inhibition, there is an accumulation on long-chain dihydroceramides. We have been found in many cancer types, from prostate, breast, lung, glioblastoma, name it. Okay, and also in tumors, in preclinical models, this accumulation. We also have proven that accumulation of long-chain dihydroceramides induce ER stress and cytotoxic UPR and cytotoxic autophagy. So validating the mechanism of action of ABTL. And the idea is that cancer cells that normally they show elevated ER stress because they have all this pressure, you know, from the organisms, ABTL is induced an increase of ER stress, and this increase trespasses the threshold which induces cancer cell death. Whereas in healthy cells, this is our hypothesis, but it has been proven certain, where in healthy cells, ER stress also activates ER stress, but in a way that don't trespass that threshold. So that's why it's a safe, let's say that it's a safe drug for healthy cells. I hope you understand the thing. So basically, 3 years ago, we finished the clinical phase 1, phase in human, and basically we found that we expected this ABTL is a very well-tolerated, orally tolerated compound. Most of the adverse events were gastrointestinal, grade 1 and 2, basically diarrhea and some problems with nausea. And it's an extremely safe compound, you know, which is given up to 4 grams a day, which is now the doses. It's like a fillet. There's a lot of drug in there. Okay, so now in the clinical phases, it's given in combination with chemotherapy, normally taxols and platins, and to improve the efficacy of the chemotherapy. So also, my laboratory, also during these preclinical and clinical phases, we develop a test to produce, to obtain pharmacodynamic markers for the phase 2B, which is now currently. And basically, we're able to measure from all these proteins for the UPR, which is activated in induced cell death, like CHOP, TRP1, 84, we're able to measure the mRNA levels in blood, okay? And we found that here on the graph, on the left half, the levels of TRP1 or CHOP1 in 25, 23 patients. At 0 means before starting the treatment, 8 hours after the ABTL was given to the patients, and 1 week and 3 weeks after the product. At 7 days is when the patient should start with the chemotherapy, and they will prolong with that. So as you can see in both graphs, ABTL induced in plasma the levels of these mRNAs from TRP and CHOP, and also we have other ones, and it's sustained even after giving the chemotherapy. So this is the first time that any UPR gene, UPR mRNA, is validated as a pharmacodynamic marker in plasma.
Miguel,
yeah.
You have 1 minute left.
Okay, quickly. And now this product we just finished a small clinical phase 2, it's called Indolang, for treating patients with advanced endometrial and non-small cell cancer, lung cancer, in combination with paclitaxel and carboplatin. And this was performed in many hospitals in different countries, especially in Rio here. And this was supported by the preclinical data. You here in both graphs, you can show that a combination of ABTL plus this chemotherapy regime induce a more robust reduction of tumor growth. So these are the results from this small clinical phase 2. And this is for endometrial cancer. It was led here by the bio by Dr. Anna Nick. And probably I have no time. The most important thing I want to reinforce is that the overall survival was like 27.5 months, which is historical with this chemotherapy. This is the chemotherapy plus ABTL, which is historical for this chemotherapy. Platinum and Taxol is about 24 months. But I think it's more impressive the results for this type of non-small cell lung cancer, which is the squamous one, in which the overall survival was reported to be 25.5 months, which almost double the historical one. These are small, they were 38 and 25 patients. We are publishing now the results of these clinical trials, phase 2, but it's encouraging. And just for you to know that ABTL is also in clinical trial now started a clinical trial for pediatric, especially neuroblastoma cancer. A work and a trial which is led by the VHIR and by Dr. Lucas Moreno and Miguel Seguno. And finally, to finish, just to show that ABTL is currently now in clinical phase 2B for the treatment of advanced metastatic pancreatic cancer in combination with the standard chemotherapy, especially in this case, FOLFIRINOX. And here are the preclinical results supporting our notion. On the left, you see in models of tumor xenografts of pancreatic cancer that ABTL increased the efficacy of paclitaxel and gemcitabine, which is one of the two regimes used for pancreatic cancer. Or below, you have the graph showing the ABTL potentiates the anti-cancer action of FOLFIRINOX, both in xenografts, but also in syngeneic tumors. In syngeneic mice, we have seen also that ABTL induces immune stimulation. It seems that it stimulates or activates the differentiation of macrophage to M1 antitumoral phenotype while downloading M2. So this is now in clinical trials in different countries and many hospitals. We so far has been collected about 60% of the expected patients, and we hope the results to get the results in next summer. I hope they're okay. So thank you.